Start recording. All right. A very, very warm welcome to everyone joining in from around the world. This is another one of our astrobiology webinar episodes from Center of Excellence Astrobiology at Amity University in Mumbai. And we are very, very pleased to have with us Ms. Anushree Srivastava, who's from India and is currently pursuing her doctoral studies at the Open University in UK. Anushree has been a friend since 2015. We go back since we started working towards understanding how we can develop a program for the exploration of Ladakh as a Mars analog site in India. And she's also working with the Mars Society, with Blue Marble Space, and as part of that, the Astrobiology India initiative. So we're very well connected in terms of many of the projects we're running, and we're delighted to have her with us to talk about salt-loving life that we could potentially find on Mars, or the proof of its past existence. So I'll just throw in a few lines, and then I'll let Anushree talk a bit about herself as well. Anushree is originally from Lucknow in India and is currently based in England. Her educational journey has started off as a student of literature, history, and music, which culminated into obtaining a professional training in Indian classical vocal music, a Bachelor of Arts in Ancient Indian History and Anthropology, and a Master of Arts in Anthropology. But while specializing in its biological branch, just out of curiosity, it exposed her to the adventure of exploration. During her graduate specialization in biological anthropology, she studied the dental morphology and its variation among the human population. Anushree is passionate about astrobiology and space exploration. Her penchant for astrobiology and space exploration largely stems from her awareness to be awed by the wonder of life and her intrinsic desire to understand humanity's place in the universe in a broader sense. To pursue her interest in the prospect of life beyond Earth, which was challenging for an art student, she joined the Master of Science program in biotechnology at the University of Essex in UK with Dr. Terry McGenity. She conducted a geomicrobiological study of over 6 million-year-old halite crystals that deposited as a result of Messinian salinity crisis in the Mediterranean region. She is particularly interested in the biogeographical and astrobiological perspective of the Messinian halite. Her research interests include understanding the prolonged survival potential of Halobacterium entombed inside halite crystals and how this understanding can be translated into prospecting similar life in Martian chloride deposits. So I'm intrigued and curious and excited to have Anushree talk to us. And we also have Dr. Jonathan Clark, who's overseeing and who will be around, and hopefully he can also help us out with some of the questions that we'll be getting. So Anushree, what we will do is we'll allow you to speak, and we'll hold all the questions until the end of your talk.
Yeah.
We'll do the question answer session there.
Yeah.
So over to you.
Thank you, Siddharth. You just talked about my entire presentation today in short. Thank you, such a wonderful introduction, and thank you everybody for joining me. Yeah, so like Siddharth said, I'm Anushree and I'm a PhD student at the Open University. I'm also working with astrobiology group at the Open University. So today I'm going to talk about why salts are so important in terms of search for life on Mars. And I'm also going to talk about my personal journey along with the journey of understanding the long-term preservation of biosignatures within ancient salts. What happened? So first, little introduction about how do salts form, how do evaporites form. So what happens is that under extreme hot conditions, the seawater gradually evaporates and leaves behind thick deposits of dissolved minerals. But it doesn't happen so straightforward. It happens in a sequence. So as water evaporates, the concentration of water keeps increasing. So first in the sequence deposits carbonate salts, then sulfate salts, and then chloride salts. So this is very interesting that if we find some salt deposit somewhere, we can always tell that what the concentration of the start chemistry of this of the diluted water would have been, whether the original water would have been so diluted or concentrated, because we see different kind of fractionation in terms of salt deposition. So while salt, the water is evaporating, these tiny liquid and gas bubbles get trapped within the crystallizing salts, which are called fluid inclusions. So I'll tell you in a minute that why fluid inclusions are important in terms of astrobiology research, in terms of preservation of microorganisms. So this is a mind-boggling article that I came across back in 2014. And it was written by Dr. Terry McGanity, who became my master's supervisor later on. So this article was about Halobacterium Salinarum, which is also considered as halophilic superhero because it's extreme salt-loving microorganisms, and it can live under highly saline environment. So this nice schematic here is showing how such halophilic microorganisms can be entombed, can be preserved within sodium chloride crystals, within the fluid inclusions inside the sodium chloride crystals, and that they can live for over 1,000 or possibly millions of years. And these crystals are also important because they provide a kind of protective environment from outside radiation. So this is very important here. And that's why we hypothesize that maybe on Mars, if there was any microorganisms living in the past environment, in the water bodies, if that evaporated and crystallized with like a salt, they could be preserved within the microenvironment of these crystals. So these are real examples of Earth's sodium chloride crystal that we found in various places on Earth. So these are sodium chloride crystals basically, and you can see this orangish, yellowish, reddish spots here. These are prokaryote, these are microorganisms. They have been entombed within the sodium chloride crystals, within the fluid inclusions. So the pictures that I'm showing here, they are as old as about 36,000 years to a couple of million years. So this is very fascinating that microbes do get entombed within a salt crystals, and they can be preserved for a really long period of time. So the red and orangish coating that you see here are actually some pigmentations that these microbes secrete. This pigmentation is called beta carotene, and they secrete this pigment in response to the highly saline environment and sometimes radiation as well. So I mean, this is very fascinating because such microorganisms have such coping mechanism that can allow them to be living covered within such salt crystals, within such ancient salt crystals. So while Mars is important because Earth and Mars share both geological and hydrological history, and Mars was habitable when life arose on Earth. So here in the red box, we see the blue color is indicating the availability of liquid water on the surface of both the planets. So this is important that we clearly see here when Earth has its first life form, which are considered, maybe they would have been thermophile, they would have been microbes that could live in highly extreme hot conditions. So in this image, we see that when the first life arose on Earth about 3.8 to 4 billion years ago, Mars had ample water on its surface. So this is very important because, and that's why we hypothesize that if Earth at that time could arose the first life form, why couldn't it be Mars? So that's why it's important. And also, I've just shown these images here that are geomorphic features on the surface of Mars that are kind of screaming that Mars once had liquid water on its surface, or liquid water once flowed about 4 billion years ago on its surface. These are gully channels. These are carvings of flowing water. And these are important indications that Mars was wet in its past about 4 billion years ago. So salts on Mars. Mars is a salty planet. So various robotic and remote sensing missions have found different kind of salts on the surface of Mars, be it carbonates, sulfates, and chlorides. The two images I'm showing here, the first one, the upper one, is the image of southern hemisphere of Mars, this region called Terra Sirenum. So all the black spots that you see here are the extent of sodium chloride salts in the southern hemisphere of Mars. So you can see that it's so abundant on the surface of Mars. And the image below, we see here are calcium sulfate salts. And this image is from Gale crater Mars. So we see that there's a very dense network of calcium sulfate salts here on the surface. So this is a fascinating animation of brine, which is salty water evaporation model in the Gale crater Mars. So what we see here, that the thin stream of water from Mount Sharp coming down to the floor of the Gale crater. And this would have happened in a cyclic manner, like once the crater was filled with water and then evaporated and then filled again. So this would have happened in a very frequent manner. And that's the question that we ask, that when this water evaporates, it leaves behind a highly concentrated small ponds of water. And this water would have been so dense with the concentration of various salts. And the question we ask, that can microbes survive in such a high concentration? And we see so many examples on Earth, Earth microorganisms, which is salt-loving microorganisms. They survive in really high concentration of salts. So this is very interesting to me personally, because I'm working towards this question during my PhD. Okay, so this is the first project that I was working on as a master's student at the University of Essex, and it was about the Messinian salt deposits. So what happened about 6 million years old, that the Mediterranean Sea got blocked off from Atlantic Ocean, and all the water got evaporated and deposited. The salt was deposited in 3 mines of Italy called Tuscany, Racalmuto, and Rialmonte. So during my master's study, my samples came from all 3 mines, and I was studying, these salty deposits from the astrobiological perspective. So my objective for this project was detection of living or fossilized microbes within the Messinian sodium chloride crystals. And so whenever we study such ancient samples, especially salt samples, we always sterilize the surface of the crystal, because we don't want any contamination. If we want to prove that the life signatures that we have detected from this crystal had to be from inside the crystal, not from the outside, because then we cannot prove that these are ancient life signatures that we found. So we always prefer to get the core of the sample, the crystal, from right inside the big chunk of crystals. We always, prioritize the core of the crystal. So we always sterilize the surface, and then what I did, I extracted DNA directly from the surface sterilized crystals. But guess what happened? My results got negative. And I really cried, because I raised my expectations so high from these samples that definitely I should see some ancient biosignatures within these crystals. But we had a plan B also. So what I wanted to test, that can halophilic microorganisms or halophilic algae, if they come together within a crystal, so their symbiosis can favor the survival over geologic times. So there's this hypothesis that algae, the green algae, secretes glycerol that provides some energy source to their fellow Haloarchaea, and that's how Haloarchaea survives, by eating the glycerol secreted by green algae. So what I did, this was completely an experimental work that I did during my master's project. So what I did, I prepared cell suspensions of green algae and Haloarchaea both. I mixed those cell suspensions. I made a sodium chloride brine. I mixed the microbial cells within the brine, and I evaporated the brine inside the lab, and I precipitated the sodium chloride crystals. And guess what? These crystals had cells inside. So so just to test that, can they be alive by getting on tomb with these crystals? So I took some crystals. So this project was running for 3 months. It was just a 3 months project. It was very short time, to be able to tell something significant. So what I did, I took the crystals which had cells inside. I redissolved them in the ambient environment, like I was mimicking their natural environment. So I redissolved these crystals, and I found out that yes, with and without green algae and Haloarchaea. So without green algae, they live for short time. But if you onto green algae with the Haloarchaea, they kind of being encased within the salt crystal. They can survive for a long period of time with the green algae cells. So this was interesting, but it needs some more study before we get to a definite conclusion. So it was, like I said, it was 3 months project for my master's study, but we are going forward towards that. And yeah, soon we will have some more information on this. Okay, so while I was just about to finish my master's project, I got this opportunity to attend Mars 160 Twin Desert Arctic Analog mission. It was a long-term simulation for human exploration of Mars, and we attended it from 2016 to 2017. And it was a pure international crew from Japan, France, United States, India, Australia, and Russia. Dr. John Clark is here, so he was also part of our team, and he was so amazing. He was a crew geologist. I was a crew biologist. So we had such a great time during the expedition. The patch you see in the right is our mission patch, and I was so excited because the India's flag was there because of my participation. But that was really a moment of pride for me. So the first chapter started at Mars Desert Research Station in Utah, USA. As you can see, this is the entire campus of MDRS. So this mission was basically divided into 2 chapters. The first chapter was at the Mars Desert Research Station. So the entire simulation period was about 3 months. And what is important about this site is the landscape belongs to the Jurassic period, which is about 200 million years old. So we did collect a lot of samples from different locations, being in the simulation, wearing the space suit and going outside. So whenever we would go outside, we'd wear the space suit. We did all the field work, all the studies in the space suit. So that was another component of the simulation of the entire science mission. The second chapter started at Flashline Mars Arctic Research Station, which is based on the Devon Island, and the simulation period was 2 months. So I would say that the Devon Island is the largest uninhabited island, and we were 6 people on the entire island. So we really did some great work there, and I'll tell you in some next slides. So Mars 160 mission was a pure science mission. I joined the mission as a Mars-based scientist in coordination with Earth-based scientists. So this was another very interesting facet of the mission, because I was supposed to work in coordination with my Earth-based scientists so that we could simulate the asynchronous communication, how if a scientist goes to Mars, I mean, how they can coordinate with Earth-based team. So this was another aspect of the simulation. So while we were there on Devon Island, we got the opportunity to access Hot and Impact Crater, which is about 39 million years old. So the diameter of the crater is 24 kilometers. And the objective, as a Mars-based scientist, my work revolved around a detection of living or fossilized microbes within the hot and sulfate salts. So the image in the left you see is the mound of calcium sulfate salt. It was so fascinating that Dr. John Clark and I went there, and we found out this location, and we were so excited because calcium sulfate salts were everywhere, and I started calling it gypsum land. So calcium sulfate was also called gypsum, which is hydrated calcium sulfate. So yeah, so why this site is important from astrobiology perspective, because of its similarity to Endeavour crater on Mars. Both are impact craters, and both had lake in its past. Both the craters had lake in the past. So and the water evaporated and it's deposited as you know, these veins of calcium sulfate salts. So it'll be interesting to see if we find any entombed, even if any organic matter within the salt crystals. That'll be really relevant from the astrobiological perspective. So we collected samples from these two locations within the crater. So these 7 samples that we collected, we did some thin section and short wavelength infrared spectroscopic analysis. John was so helpful for me, you know, to be able to carry out this work. So we found out that sample is mainly composed of gypsum, like I said, the hydrated calcium sulfate salts. But we also suspect that some organic matter, which is the indicative of previous life that would have been living in that environment before. We suspect that we have some organics attached with the matrix of the crystal. So to be able to confirm that the GCMS analysis, which is gas chromatography and mass spectrometry, this analysis is still underway, and we are still in the process of, you know, getting the entire the results out. So hopefully we see some organics with the samples, within the samples, which is about 40 million years old. So that'll be really fascinating if we, you know, come up with some definite conclusion. So the next chapter in my entire research career regarding this life in saline environment was mine analog research, the chapter 5. This expedition was held in Bulby Mine in England, which is north of England. And what is fascinating about this site is we have this 1.1 kilometer below the ground. We have this astrobiology lab, and people go there and then do some really cool science. So this happened for the first time that we were 36 people inside the mine. So this is a record that we made at that time. So this expedition was held in about 2017. So the project I was involved with was about Raman Spectroscopy, and we wanted to do some mineral identification and live detection work. Okay, I was hearing some voices. Okay. So yeah, so I was saying that the project I was involved with was Raman Spectroscopy, and we wanted to do some mineral characterization work and live detection. So this site is important because these are Permian salt. These are about 250 million years old salt samples. So Bulby Mine has all kinds of salt. It has sulfates, it has sodium chloride, it has potassium chloride, it has some really complex mixture of salts also there. So these are really old. This is 250 million years old salts, and we wanted to test Raman spectroscopy to identify the mineral composition of various ancient evaporite minerals, and to be able to test any buried life signatures from different locations in the Bulby Mine. So we recently published this paper actually in 2018. We published the results of this work. And for Raman spectroscopy, we found that sodium chloride is really hard to identify with Raman because it's a transparent crystal, and it's almost featureless. So you can see the graph here in the below, that sodium chloride, that is halite. We don't see any significant peak here. But for sulfate, it gave us a really good peaks to be able to identify the mineral composition. So one thing was the halite. One thing was halite was hard to identify, and we did not find any organics within those crystals using Raman spectroscopy, because I was working only on this component. And we also found out that we need to test, several other instrumentation in terms of, to be able to get a definitive results of for any whether it's a mineral identification or life detection. So it's not just one technique is not enough. We need, a coordination of several other techniques to do this work. So even though during MINAR 5, we did not find any organics within the crystals of 250 million years old, but in a very recent paper by Charles Cockell, they found out lipid biomarkers within such ancient sodium chloride crystals, which was really, really fascinating. So what are lipid biomarkers? So we know that the microbial cells or any cells are composed with a cell membrane. So lipids are the fundamental component of the cell membranes, and cell membranes are made of kind of bilayers. There's 2 layers, and each component of these bilayers are called a phospholipid molecule. So this is that's why lipids are so important, because these are fundamental feature of all life we know. So the phospholipid molecule has 2 segments. One is called the phosphate head, and the second segment is called fatty acid chain. So just to let you all know that the phosphate head is very much prone to degradation in environmental pressure, but fatty acid chains are really hardy and robust. They can be preserved over geological time. So that's why we focus on fatty acid component of the phospholipid molecule. And this paper found out that fatty acids have been preserved in 250 million years old sodium chloride crystal. That suggests that organic matter can be preserved within the evaporites, and they can be as old as the crystal itself is, which means that we can find indigenous organic matter within such ancient minerals. And this also tells us about why we can hypothesize that if on Earth we can find such examples, definitely when Mars has lots of salt on its surface, we have the possibility to be able to find lipid components. If there was a life about 4 billion years old, when Mars was so compatible for any kind of microbial life, we can be hopeful that maybe we can find some really hardy features features of life that can be preserved over such a long period of time. So that's why I say that it has a lot of potential to find such biomarkers, especially lipid biomarkers within the salt deposits of Mars. So just going towards my PhD now, since we are talking about lipids, I'm kind of asking the same question, that whether sulfate minerals can act as a viable substrates for a long-term preservation of lipids, and especially under present Mars conditions, which has lots of radiation. The surface is big with high doses of radiation, and it's low pressure, it's really cold, it's arid. So under such conditions, can we still find these lipid molecules attached with the crystal, with the salt crystals? And the second question is whether these signatures can be detected, can be explicitly detected. So these are 2 main questions that I'm asking through my PhD. So we've gone to different kind of natural environments. We've collected samples from all those environments. But during my PhD, what I'm doing, I'm creating my own samples to be able to do a very controlled experimental work in which we know everything. And in this condition, it's a lot more easier to be able to get a definitive results. So what I'm doing, I'm creating a brine, which is also called salty water, which is also called brine. So I'm kind of creating that brine, which will be similar to the Gale crater brine, the lake that we suppose that would have been present in the Gale crater in the past. So I'm creating that brine. I am going to put microbes, selected microbes within the brine. I am going to evaporate that brine under Mars conditions. And then I'm precipitating the salt crystals, and the salt crystals are going to be with this microbial cells. So this is a very controlled experiment that I'm doing during my PhD. So my question, like I said, my question is, can those salts, which has the authentic chemistry similar to the Gale crater brine on Mars, can we detect such lipid signatures within those crystals? So this is the basic question that I'm asking through my PhD. So while I'm talking about the detection of those signatures, which is another very important component of this entire lab detection program in astrobiology, I must mention sample analysis at Mars, SAM instrument suite on board NASA's Curiosity rover. So SAM is right here in the belly of Curiosity rover. It has three main components: tunable laser spectrometer, quadrupole mass spectrometer, and gas chromatography. So I'm not going to go into much detail, but just to give you a little overview of what these components do. So first of all, pyrolysis. When a Martian sample is introduced to the sample analysis at Mars instrument, it is pyrolyzed first. So what pyrolysis is, when a sample is decomposed after applying extreme hot temperature, and that's decomposed, and the gases evolved from that decomposition are sent to the gas chromatography column, and where these molecules, the complex mixture of organics, are separated. And those separated organics are sent to the mass spectrometer to be able to detect it. So this is the focal functions that these three components do. But SAM also has an additional component, which is derivatization. So why we do derivatization is because when some really complex organics cannot be detected just by GCMS, we apply some chemical reagents. In Curiosity, we have two chemical reagents, MTV, and PMAH. So what these chemical reagents do, they make those organics more volatile in a more gaseous forms, so that they can be easily detected through this instrument. So what SAM is basically doing is looking for organic molecules on the Martian surface through pyrolysis GCMS. And like I said, it also does derivatization works just to be able to detect some really complex organics within the Martian sample. So the first derivatization experiment was done in December 2017, and I was so fortunate to be able to join the team to interpret those results. So why I'm talking about this GCMS component, because through my PhD, I'm also kind of testing this technique on my own samples, that how compatible this technique is to be able to detect a complex organics or lipid biomarkers, especially using pyrolysis GCMS. And I'm also testing whether these salts are the compatible substrates to be able to detect such signatures. So while we are talking about the GCMS component of Curiosity rover, so this is the proposed route of NASA's Curiosity rover. So the rover is currently in a green zone here we see, and is going towards the yellow zone, which has a lot of sulfate salts. So this is very exciting for my work, because the rover is going towards the salt now, and it's going to test organic molecules, the detection of organic molecules within the sulfate salts. So this is very timely. My PhD research and the Curiosity's work is very timely and relevant at this time. And I'm really looking forward what comes out. Just to mention that, like I mentioned, that Curiosity rover does the sample analysis smart instrument suite does some derivatization work. So I'm also testing how this derivatization works on the salt samples. So I think it's going to be, like I mentioned, that it's going to be very, very timely. What I get, with the results I get of my PhD work, and how the curiosity is going to explore sulfate bearing unit, and what the results come out from both the works, and how we can, how my work can inform the mission itself. Yeah. So like I just indicated this yellow zone here. So this is the list that I've mentioned here of all the organics that MSL SPAM has detected. I don't know what is this. Okay, so so these are the organics that SAM has detected so far, but we still don't know the sources of these organics. It could be biotic, could be abiotic. So that'll be really interesting to know that how these organics came from, where these organics came from within the samples. So yeah. So the major takeaways are that Martian salt deposits are potential substrates for lipid preservation. We have seen, so we've seen really recent paper that lipids can be preserved in 250 million years old salts. And also the robotic exploration and sample return missions are kind of prioritizing salt crystals for the live detection program, for live detection experiments. And we also need more experimental work to better constrain the preservation of biosignatures and degradation also within Martian salts. Because like I've mentioned that these salts are on the surface of Mars, and the surface has been bathed with high doses of radiation for such a long period of time, about 3.7 billion years or maybe 3 billion years. But this is a really long time for any organic matter to decompose and degrade. So we need more experimental work where we expose such samples to the really harsh environment of present day Mars, so that we can say what the degree of degradation, can we really detect those organics in the salts. So this is the big question for me. And I also found out during this entire, my whole regime of work is that it's not just one technique. We need a set of spectroscopic technique to be able to detect life within the Martian substrate. Like I said, it's been really difficult conditions from past 3 billion years old on the surface of Mars, and we need a set of techniques. We need to test different techniques that can give us explicit signals of organic matter preservation within different salt crystals. Yeah, and my journey goes on. That's it for me. Thank you, and I will take any questions.
Thank you so much, Anushree. Sorry for the disturbances in the middle.
That's fine. I was kind of, what's happening? Somebody's saying something.
Yeah. Well, we have the option of either doing a webinar mode or meeting like this. I think for the next meetings, we'll have to probably shift to webinar mode because there are people joining in from everywhere. And we try to keep a track of who's trying to disrupt the meeting. So I've locked the meeting so nobody else can join. And I've also ensured that no one can unmute their mic.
Yeah.
So let's just quickly take some questions. There have been a few, and we'll try to take them all. But let's see from the one right from the start. Let's see.
Yeah. Should I? Okay. So are you gonna speak out the questions?
I will. I'll be speaking out the questions to you, and you can also read the chat. So the first question is. Okay. Let's see. So Gaurav Yadav asks. How can we detect a possible lipid biomarker using Raman spectroscopy? How can we select the wavelength range which can detect the lipids?
So so to be able to simulate the wavelength, I mean, Raman is going to be used on Mars 2020, and the wavelength I remember is 500 something. So I mean, you need to see which labs have at similar wavelengths, Raman spectroscopy with similar wavelengths, so you can bring your sample there and you can test under similar conditions. So yeah, so wavelength is just about matching. Yeah, like at Open University we have Raman with the not exact wavelength, but it's about 400 something. So yeah, I mean, just to test, you need to find a lab which has the similar facility that we have on board Mars 2020.
Mm-hmm.
And for lipid, it depends. I mean, that's still a question. And that's the question that I'm asking through my work also. Can we detect? Can we detect such signatures? But lipids are usually considered a hardy components, and that's why people are focusing on lipids. Like we see in a recent paper by Charles Coquel that they have found free fatty acid, that the second component of lipid biomarkers within 250 million years old. That's a big deal. Mm-hmm. If they can explicitly detect such such biomarkers in such ancient sodium chloride crystals, that's a big deal for me. So yeah, it's still a question. You need to test, mm-hmm. And that's also I'm testing through my PhD work.
Okay, thank you. So a question from Payal Agarwal. I'm not entirely sure what she is referring to. So Payal, perhaps rephrase your question and pose it again below. I'll get to it later. She's asking, what is the 3x3 cube thing next to the rock samples with colorful spheres within? Does that strike a bell, or should we ask her to clarify?
No, please read one of the slides. Can you ask her? I mean, I don't know which slide she's referring to.
Okay, so Payal, could you please indicate the slide number that you're referring this to? And I also ask and request the other questioners to indicate the slide number.
Maybe this one? I think this one she's referring to.
Might be. I'm not sure. Let's wait for her to confirm, and then we'll get back to her question. Okay. So Pradnya asks, what tools did you use to plot and analyze the data that you've shown in your slides? What are your software tools, the ones that you're using?
Which one?
Okay, maybe just in general, like, because there are a lot of students joining in, so you can, maybe it'll be useful for them to also know, what kind of tools are you using for your data plotting.
Well, the software that I've used is Origin Pro. When I was working at NASA Goddard, I was using only Origin Pro for plotting my data. So yeah, Origin Pro, and some people are also use R. Like the Open University, we use R software a lot for any statistics and data analysis.
Mm-hmm.
Yeah.
Perfect. There's a comment from Logan McTeer. Be careful of false positive results for non-selective detection analysis, such as spectro.
Yeah, I mean, yes, of course.
Okay.
Yeah.
Next question is from Kavaljeet Kaur. Are there as there are many halophiles present, on what basis will you choose these microorganisms for the experiments?
Well, that's a good question. On Mars, it could be anything. It could be anything, but we start from somewhere. Like the potential microorganisms, I would say, of course, halophiles that love salty environments, also methanogens. Methanogens also a very potential type of microorganisms that they also carry a very ancient way of metabolism, which is hydrogen oxidation and reduction of carbon dioxide. So if you talk about the first life on Earth, what I've seen that it could be methanogens and thermophiles also. There are a lot of organisms that live in highly extremely hot environments. So there's a lot of papers that argue about that the thermophiles were the first ones that arose on Earth. So there are several. Like I said, it could be anything. So you just need to start from somewhere. Like for me, I'm still in the process of selection. I have not selected the potential microbes for my PhD work, but it could be anything. You just go there, and the potential ones are methanogens. I would say methanogens and halophiles, thermophiles. Yeah.
All right, thanks. Next question is from Prachi Mayekar. She asks, how can you tell what was the composition of the liquid inside the crater? I think I'm not sure which crater she's referring to, but maybe a generic.
Okay, so okay, yeah, I understand that. So we have a paper so there's an astrobiologist, basically mineralogist, geologist in our group. She's Dr. Susanna Swenger. So she has a paper in which she has modeled the brine. She has modeled the composition of the Gale crater brine. So my work is focusing on that modeling. So she has given me a composition. So I'm working based on that composition. That's the authentic composition that she has modeled. That's a prediction that we had this mineral composition in the Gale crater brine. So yeah, and she has also predicted that this brine would precipitate the calcium sulfate veins that we see in the Gale crater. So that's the entire idea of the modeling.
And does this model also incorporate some of the observation data that Curiosity has?
Yeah. So that's consistent with the data that we have through rovers and the orbiters.
Perfect. Thank you. Next question is from Shrabani Patnaik. Shouldn't we switch our target focus to Tharsis Plateau, which was geologically active once, because there is a chance that we can find preserved ancient life forms in Martian lava tubes, and also dried salt beds nearby a volcanic plain could serve as a great location to look for salt metabolizing primitive life.
Yeah, that could be a possibility. That could be a possibility, and someone needs to explore that. Yeah, of course.
Mm-hmm. Yeah. And another question from Srishma: can we simulate aging like billions of years in controlled laboratory simulation experiments?
Well, aging billions of years is not possible. For that, I think you'll need to do a chain experiment. But at least, if you do a year of experiment, like if you keep your microbes entombed within a salt crystal for a year, maybe it can give you some sort of indication of their capability to survive, being encased within such a microhabitat. So yeah, I would say it can give us some indication, but for simulating the billion years old, that time scale, I don't think it's possible in any of the labs. Yeah.
It'll mainly be a modeling effort.
Yeah, the modeling effort, and I think you can maybe increase the power of radiation. Like you can increase the magnitude of radiation. To be able to simulate such a long period of time of exposure of radiation and harsh present day Martian conditions. That's just I'm thinking of loud. Yeah. Because I don't think that such a long period can be simulated in a lab. Yeah.
Thank you.
And also there's one limitation that if you're working as a PhD student, you have a limited time frame, So especially UK PhDs are 3 to 4 years, which I feel is so less for a, yeah.
You wish you had more time?
I wish I had more time. I mean, it's for me, honestly, it's an endless process. We get a degree, but the question still remains.
Yeah, that's true. Okay, so another question from Kawaljeet Kaur. While evaporating the brines for forming of salt crystals, doesn't the heat cause damage to the bacteria?
No. I mean, that's the question, because when a microbe gets sheltered within a crystal, the crystal itself provides a protective environment from all kind of austere extremity. So that's the advantage of why we explore salts and their entombment of microorganisms within the salt. So I don't think heat is going to do anything because microbes are already sheltered. Yeah.
Okay, thank you. Next question is from Mohammed Wahidi. He asks, did you simulate the process using algae and its symbiotic bacteria because you were speculating that it happened this way on Mars when it was habitable, or just because it was easy to preserve in salt crystals and then extract later on?
Both the things are true. Both the facts are true. because there could be some algae, but yeah. So when I was doing my master's, the focus was not basically Mars. It was just understanding the mechanism of why even on Earth, such microbes can live for such a long period of time. Because there are papers, I mean, you can go and check online. There are papers that say that the glycerol that green algae secrete, it helps halophile to take up that as a food and be be viable for a long period of time within a salt crystal. So when I did that experiment, I was not entirely focusing Mars. It was a too early stage for me, so I was just focusing on Earth microbes. But that's also kind of compatible research question for a live detection way of Mars. Yeah, I would say.
Awesome. All right. We have like questions coming, so I hope you're okay with me going through.
Yeah, that's completely fine. Yeah, just go ahead.
So another question is,
good to have a discussion.
Yeah, I agree. Another question is, what biosensors are used for detection of biomolecules in Mars-like places? I want to know the biosensors used on Mars rovers.
I am not entirely aware of biosensors, but I have come across several papers on that. But I'm sorry, I'm not entirely sure the technique involving biosensors.
I think primarily we're looking at signatures of consequences of biological activity. So we're looking in the form of alteration of minerals. You're looking at physical structures that are formed, just like some of the structures you showed us that could have been formed because of life. So I think those are right now the kind of instruments that are there on
So predominantly the instrumentation that we are sending on Mars is based on a spectroscopy. That are actually doing some wet chemistry experiments there on the Martian samples. But there's another very fascinating component is sample return mission, which Mars 2020 is going to approach. So that's going to give us advantages of doing our own, the work that we are testing here, the techniques that we are testing here, that what are the best techniques that that are compatible for live detection. So that's going to be an advantage that we're going to get the sample and we're going to use our own techniques here, the methods here. So I think, if that answers your question.
Next question is from Srividya Subramaniam. She says, thank you for the amazing talk, Ms. Anushree. My question is related to your master's project. What kind of controls did you set up for your experiment? You mentioned Halobacterium algae symbiosis via glycerol secreted by algae. Did you try using pure glycerol, any other mixtures? What is your take on any such variations?
No, I did not use any glycerol because that was not my question. If I would apply outside sources, that would have ruined my experiment. I wanted to test whether naturally it happens or not. Whether green algae, secretes the glycerol and Halobacterium takes it up as a food source. So I did not want to apply any outside source. I wanted to, happen it naturally so that we can test this hypothesis.
Okay. Next question is, why halide is not detectable in Raman spectroscopy? Why are the peaks not shown in the detection?
Because halide is a transparent crystal, compared to other substrates. Because we also tested a mix of sulfate salt, and that gave us really good, three, like I also I've shown here, 2-3 peaks, so that we can easily identify the crystals. But halide was so transparent, so that if you shoot the beam, it just goes through. It does not reflect because it's transparent.
Awesome.
Yeah, so like I also mentioned, it's featureless. It's transparent crystal.
So,
another
question. Sorry.
Yeah, just go ahead.
Another question from Mohammed Vahidi is, wouldn't you risk the damage of the salt crystals during the sterilization process?
No, and that's what we want to do. We want to damage the outside contamination, but not the inside.
Okay, perfect.
Yeah.
So Payal has clarified on her question regarding those 3x3 cubes. She says, I'm referring to the slide on Minar 5, the Raman Spectroscopy, slide number 18, probably.
Mhm. So which cubes?
I think she's talking
So
about
these are scales.
yeah.
These are actually, I took this picture of one of the instrument that we tested, Clupy. It was an imaging instrument. So I just took up this picture from those results. So these are sort of scales, a color calibration, I guess. Yeah, but that's also working as a scale. I put it just for the purpose of a scale.
But could you elaborate on why do you need color for
I
calibration?
cannot, because that was based on the Clupy instrument,
So the
and
instrument
I'm not
requires,
entirely sure. Yeah.
okay. So the instrument requires to calibrate
Yeah.
itself based on the different shades,
Yeah.
the wavelengths
Yes.
that are coming
Yeah.
back, and then
Yeah.
informing
It's
it.
just a coloring calibration, something like that.
Yeah. Okay. Question from Anurup. Did you consider perchlorates in your study?
No, not yet. Actually,
Okay.
the problem is that in our lab, we are not allowed to use perchlorates because it's really hazardous,
Yeah.
toxic. I tried initially. I tried to convince them, but I I could not.
Okay.
Yeah.
Question from Yusuf Salam. How can you eliminate the problem of the secondary formation of fluid inclusions and the problem of
Yeah.
contaminations, not in the lab, but through the geological time? And how could you affirm that biomarkers detected have the same rock age?
Yes, that's a big question. That's the big question in this area of research. So secondary formation, that's where geologists come. You know, they can know the history of 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 any kind of redissolution and reprecipitation of salt. Like if I give this example of Houghton impact crater, so no, not a Houghton impact crater, but this another paper I came across. One of the students of my master supervisor, Dr. Terry McGanity. So one of his students worked on some salt crystal from Chile, and there was this. They knew by the fact that the lake has not been redissolved from last I think 2 million years old, 2 million years ago. So yeah, something like that. So that's like I said, that's where geologists come from. They can tell you the history of a particular site. So you know that how long it's been that the environment has not been in contact with water, so that there's no redissolution and reprecipitation. So geologists would tell you. A work of a biologist is just to grab information from geologists in terms of salty environment, and then go ahead from there. So yeah, I mean that's a big problem. And for knowing the indigenous nature of a biomarker within a salt crystal, that's why I mentioned that we always prefer the core of the crystal. We don't care about the outside cover of the crystals. We always go to the core, so that we know that it has not been in contact with any kind of redissolution and reprecipitation. So yeah, that's the approach that I take whenever I study such ancient samples.
Thanks, Anushree. We'll just take 5 last questions, and then because we're close to finishing towards the hour. So the next question is, what kind of sterilizations are used for sterilizing the surface of crystals before the study?
Okay, so the technique that I used during my master's work, it was a set of some solutions. For example, hydrochloric acid, sodium hypochlorite. These are toxic, really toxic solutions that would kill any any contamination on the surface. So you can find various papers on surface sterilization techniques online. There are a lot of papers, and one of the papers that I just mentioned, the student of my master supervisor, Grayman et al., 2011. If you check Grayman et al. 2011, it has a very rigorous technique mentioned there, that they tested for surface sterilization. And I used the same technique for my master's work. So there's I think 7, 8 solutions, and we dip crystals one by one into each solution for a certain period of time. So so the end product is totally sterilized surface crystal. So yeah, so that's how I did it during my master's work.
Thank you. All right. Next question is from Ilan Kuzali Elavarasan. What are the potentiality of fungi to be present or be introduced to salt brine for microfiltration?
Microfiltration? I'm not sure about this term. But what was the first part of the question?
What is the potentiality, or I think she means the probability of fungi to be present or to be introduced to salt brine? Yeah, I'm not very entirely sure.
I think I mean I think it depends. It depends from environment to environment.
Okay.
You know. Yeah, that's what I can say.
Okay. All right. Well, we'll just move on to the next question. When will you get the results from the rover on Mars? What are your expectations?
Which rover?
I think he probably means the Curiosity one.
Curiosity. I mean, we already have a lot of results. Like we have, like I mentioned in one of my slides, that these are the list of organics that we have detected so far. These are indicative of previous life, but it's not a surety because we still don't know the source of those organics. It could be, the source could be abiotic and biotic. But not sure. So Curiosity has already given a lot of results in terms of organics, mineralogy. But what I'm excited most about is the next sulfate bearing unit that Curiosity is going to explore, because that is compatible to my own PhD work.
Mm-hmm. Awesome. All right, next question. Anushree, great talk. Similar to fatty acids, do you expect to see other biomolecules such as amino acids and or nucleic acids in the 250 million year old salt deposits?
It could be. But why we only focus on fatty acid? Because they are very, very hardy. They're very, very hardy, and they are less prone to degradation. So that's why we focus on lipids. But amino acids, carboxylic acids, they could be detected, but they are very complex signatures also. And that's why I mentioned derivatization that Curiosity rover SAM is doing. And that's why we have this particular component with, because that's how you make these complex organics such as amino acids, carboxylic acids more compatible to be detection. They make the derivatization technique makes them more volatile, more gaseous form, and more easier to be able to detect it through through the spectroscopy.
So connected question to that is from Gaurav. Gaurav Yadav, he asks, how do you extract the lipids from the salt samples?
There are techniques, and you can find lots of papers, the extraction techniques. So the most classical example is Blyg and Dyer. So Blyg and Dyer is a classic example of extraction lipids from microorganisms directly. And I think some papers have also used this technique from extraction of fatty acids, even from rocky substrate, and I mean salt substrates maybe.
Mm-hmm. Awesome. So that's the end of the scientific questions. Perhaps I'll just pose the final question to you, Anushree. Can you give us some lasting comments for today's session on what has been your experience working in the field of astrobiology over the last few years, and what are some of your immediate targets that you wish to achieve in the next year or 2 years or so?
Well, my experience has been great. I mean, it has been fulfilling. It has been fulfilling because, like you mentioned in my introduction very initially, that I come from a very different background.
Yeah.
I come from arts. I come from social sciences. I come from anthropology. I come from music. So why I'm here is because I'm passionate about it, and because I enjoy doing it. So of course, my experience has been great. It has been very creatively satisfying, I would say. Mm-hmm. And especially when I was working at NASA Goddard, it used to be like intoxicating, I must say. Like I wouldn't wait to go to lab and start my work. I wouldn't wait to, to have a morning and go to work. So it was like that. And it has always been like that. And that's why I'm here in this area of research, having all odds, all drastic differences that I had initially. And it took me some time and a lot of hard work to get to where I am today, whatever I could do today. And what was the second part of your question? Sorry, I forgot.
No. The second part of the question was maybe like, what do you think are some of the targets? Because you're also part of Astrobiology India and Blue Marble Space and Mars Society. What, in your view, are some of the challenges that we are facing that we need to accomplish, or you personally would like to accomplish, aside from, of course, your personal PhD and your studies, for the next 1 or 2 years? What are some of the things that we should be focusing upon?
I think we should be focusing upon on spreading more awareness about the science, because I still, get questions like how to become an astrobiologist. That's the fundamental question that most of the students, ask, and that's the question that I would asking about, five years back, maybe. So I think we need to sort out this question.
Yeah.
And we need to spread enough awareness that anyone can be astrobiologist. Just anyone can be, and whoever you are an astrobiologist, because if you're a philosopher, you're a historian, whatever field you're working in, you are an astrobiologist. That's the beauty of this science, that it is so generous, it is so relevant to whatever you do in life. I think that's what I have understood about this science. So we need to spread more and more, to be able to answer this question, like how to approach this field, how to be more, into the word of, asking philosophical questions, I would say. Because people do all sorts of work, but they do not go into deep that why we are exploring whatever we are exploring. I think that's the fundamental question.
Very, very, very well put. And I
Thank you.
completely
And personally, I don't see any challenges.
Yeah.
At all.
Perfect.
Everything is possible. It's just that you need to be patient enough to stick and hold on.
Absolutely. I completely agree with you. So with this, what we will do is we will formally close today's session. I encourage students to, and also other participants, to subscribe to our website to get more details on the next webinar, whenever that takes place. We also have an ongoing astrobiology course, and we will try our level best to include those who haven't signed up to also be able to view that course. And with this, thank you very much. Thank you, Anushree, for taking time out of a busy schedule and your PhD. We wish you all the very best.
My pleasure.
All the very best with the completion of your studies, and stay safe. And we're all in this together. And thanks everybody who signed in. And more information will be released on the next events on our website. So
thank you
very much.
Thank you. Thank you so much, Siddharth.
If you could just hang with us for a minute, I'll just end the recording, and we'll wrap it up from there. Thank you.